So my name is Beatriz Serrano, and I am presenting Demonstrator 6 on behalf of the team that you can see here in this slide. I would like to start by introducing a bit the biological background of this project. Our aim was to reanalyze public images to gain insights into the nucleolus biology. Usually, the screening experiments are focused on particular biological processes, and some information is neglected simply because it's not of the interest of the study. So the phenotypes are there, information is there, but it's just not analyzed. And we wanted to have a second pass on this data and try to find something else. We pre-selected a few screens that are available in the image data resource, which is the IDR that you see there. There is plenty of data there, microscopy data. We selected in principle a number of experiments that you can see there classified as gene silencing, overexpression, and compound screens. And the type of images that you can see is like the one on the right side of the slide. This is just a DNA channel, so there are usually more than one channel. This is just a DNA staining. The absence of DNA, it's the nucleolus that you see here pointed by red arrows, and it's like a hole in the nucleus. And what we wanted to see here is if when a gene is silenced or expressed or there is a compound acting on it, if the number of nucleoli per nucleus is changing, if the shape of the nucleoli are also changing, or they distribute in a particular way within the nucleus. And just to give you a more concrete example about the phenotypes that we can find here, you see here in blue, marked in blue, the nucleoli evenly distributed in the nucleus. So they are medium size and they are all over the nucleus. But there are other cases in which the nucleolus is absent, like you see in the bottom left here, because the cell is dividing, so the chromatin is condensed and there is the nucleoli is not visible there. There are also many examples of the prominent nucleolus that you see here in green, in which there is a big one and maybe small ones also present there. Another possible phenotype is when they are distributed close to the boundaries of the nucleus, as you see in orange. So with this data, we designed a pipeline in which we first needed to access the data, then segment to extract these two objects, on one side the nucleus and on the other side the nucleolus, and perform the feature extraction and then the downstream biological analysis. There are two big blocks here. The first one is automatic, so the data access and the segmentation and feature extraction we decided to do with Galaxy. And this could be completely automated, but the second part would require more interactive work, and that's why we decided to go for notebooks. Accessing the data from Galaxy wasn't possible at that moment, so the way we did it was going to the human protein atlas. We selected all the genes that were involved in the nucleolus regulation or nucleolus in general. And then we took these genes and went to the IDR to select all the images that were related. It wasn't possible to access that, so we needed to create a Galaxy tool to download the images directly from the IDR. I will elaborate on this later a bit more. And for the second part, segmentation, a very popular tool, it's Cell Profiler. Many image analysts use this one. This is composed by different models that are performing very simple tasks, and you chain them in a pipeline, and then you can perform your whole analysis there. So here we used 22 cell profiler modules that were wrapped into 19 Galaxy tools. That was done by my colleague Ilsun from Work Package 2. And all these tools are available for people to use it. So I will now show you what were the challenges with the data access. So at the moment that we started this analysis, there was one data set that was not available. And the rest of them, we could use the images that we were selecting. There were different challenges, and we were in contact with the IDR team all the time. They were very responsive and helpful with this. But the main problem it's usually the lack of metadata standard in imaging in general. So we open a discussion on GitHub, and also open this discussion on the ImageSC forum, which is where the imaging community discuss. And the main problem is that people are submitting data to the IDR, but they all follow their own metadata vocabulary. So, for instance, we can find, for defining a control, we can find the word control, but we can also find scramble RNAi, or not target, or there are many different words for describing the same thing. So this lack of metadata standard is particularly challenging in our case, because we're integrating several experiments at the same time. And so we needed to have this in a bit more homogeneous way. Okay, so the IDR tool that I mentioned before, this is something that was triggered at the World Package 1 hackathon in Berlin, where I had the opportunity to meet Jean-Marie Burel from the IDR, and also Björn Gruning from the Freiburg Galaxy team. That triggered a discussion about accessing the data and how we could do it. And that was materialized finally in a hackathon that we organized in Freiburg last February. And the tool looks like what you can see here in the screenshot. One needs to select the image that one needs to download, so image IDs from the IDR. Then it's possible to also select what is the region of interest. In our case, we are, for instance, interested in the DNA channel of all the screens. They are, again, named differently depending on the experiment. If you have a set stack, you can also select which plane do you want to take, or if this is a time-lapse screen, which time frame you would like, the time point you would like to take, and the same for cropping the image. So in our case, we needed to crop it because the effect of the siRNA, it's mostly focused on the center of the image, so we needed to get rid of the boundaries. With this tool, you only download the particular region of interest instead of bringing the whole image back to Galaxy. And then one can download this into a tarball to avoid transferring so many small files. And this is a recent development. Maybe 1 month or two ago, there was a demand from someone from the imaging community that wanted to have access also to local images that they have in their own Homero instance. And now this is possible, so users in Galaxy can go to their user preferences, select, input the details about this Homero instance, and then here in the tool, select that you want to go to an Homero instance, local Homero instance, and that's it. You can retrieve images from there too. This is particularly useful for images that are not public. Cell profilers. So for those of you that are not familiar with Galaxy, this is how the interface look like. We integrated different tools, so you should be able to see it here. This is the IDR download, and there are many other, as I said, independent tools for each one of the modules of Cell Profiler. In the central part, one have the analysis part to tune the parameters, to design workflows, etc. On the right side, there is a history in which one can see all the steps are running, and also explore the results. So in the end, once we have all the pipeline design and all the tools integrated, this is the final workflow. We needed to do several passes of segmentation. So first, we need to identify which is where are the nuclei in the images, and then detect the hole is inside, only the ones that fall inside the nuclei. That means a second segmentation pass. And then we also wanted to get the background. This is important, especially for quality control, because that would help us to detect some low contrast images or brightness backgrounds that are not helpful for the study. So it's mostly to detect artifacts. And finally, the feature extraction would affect all of these objects that we identified before. So we wanted to have features related to the nucleus, to the nucleolus, to the background, and to the whole image. And you can see at the bottom here that there is the IDR tool, and on the right side there is the cell profiler tool. So then more specifically, one can take the image IDs that I showed before and then download everything as an image tarball that can be input directly into the cell profiler. So the way it works is all these modules that I showed before, all these Galaxy tools could be creating a pipeline that is only run at the end in this cell profiler tool that you see here. So it will take the whole pipeline, the tarball of images, and then it will produce the results. How we can run this, I will show now. So first one can get the import the workflow from the workflow hub that I will mention later, and then can explore how the workflow looks like and check that all the parameters are fine. That one is selecting the image IDs you want, and also the channel. The same for all the different tools. One can check that everything is fine and then run the workflow. It gets scheduled. And once everything is ready to run on the cloud, you can see the steps on the right side. And now we will see that the three first steps will start computing. Once we finish the computation, we get something like this. So in the center, you see how the results will be shown in the Galaxy history. And this is green, meaning that everything went well. We will get some CSV files that are related to the different objects that we've been analyzing so far. So at the level of experiment, this is data related to, for instance, the number of images that you are analyzed. It's global about the experiment. Then we have specific for the image, for the nuclei, for the nucleoli, for the background, to define the relationship between the objects, to know which nuclei goes inside which nucleus, these kind of things. And we also get some images. So like the one on the left, you probably don't see the numbers on top of each one of the nuclei, but you need to believe me, there are numbers there in red. And this is the ID of each one of the nuclei. This is just for exploration purposes. So if we find something interesting, we can go back to the image, check which nucleus we are looking at and what is the special phenotype there. And on the right side, you see the combination of the segmentation masks. So in principle, we will have both segmentation masks separate, but also one combining. This is to make sure that we are detecting black holes that are only inside the nucleus and not just random spots outside. So you see in blue the mask for the nuclei, and in magenta you see the mask for the nucleoli. And this we will get for all the images that we are analyzing. I mentioned already before the workflow hub. So this workflow is available for people to use it. I've already received some inquiries from people. Workflow hub, as you know, is an effort from Work Package 2. And now it's possible from the Galaxy interface to import this workflow directly and just click run, as you saw in the video before. There is also a tutorial available. This is the GTN, the Galaxy Training Network, under the imaging category. So there is this nucleoli segmentation and feature extraction using Cell Profiler. So anyone can take this tutorial and do it on their own, or just use it for teaching and share it with people. Here we introduce the biological background, like I've done here today, and also we detail all the steps that are needed to run this workflow with all the values for the parameters and justification why these values are important. Okay, so the impact of this project on EOS. I think that this is a bit early days to know exactly what the impact would be in terms of uptake. We provide an example of a very commonly used steps in imaging. This is a typical use case of urban imaging, and we're running in the Galaxy environment that allows other communities to access to that. So having a cell profiler available in Galaxy makes it useful for other communities that are maybe starting to add imaging into their own analysis. The same for the IDR and Omero tool, in which we are opening now this access to this data to people from different communities, not just imaging. The workflow has been published in the Workflow Hub, and this allows the reusability of the whole workflow, but it's also very easy to take it and adapt it for different needs. So, for instance, there are many problems in which one only needs to take the nucleus. They would just take the first part of the workflow and that's it. And it can be very easily adapted to many other biological problems. There is also the training material available in the Galaxy Training Network. The future work that I would like to do is to perform some workflow testing. This is something that we haven't started yet, but we would like to do. And we would also like to grab this project into a scientific publication. In terms of sustainability, this is a contribution to large open source communities, meaning that it's more likely that it can be picked up by someone if there is need. And also having the training material available, it's also helpful for people to kickstart an imaging community around Galaxy. And the interaction, I've been mentioning people already on the presentation, but more explicitly, Jean-Karim, he designed and supervised the project. Isun was the one integrating the tools into Galaxy. Jean-Marie, our expert from Work Package 1 and the contact person at the IDR, and Bjorn Gruning from Work Package 2 also as responsible for the integration into Galaxy, helping with that part. And these two experts, as I mentioned before, I met in the Work Package 1 hackathon in Berlin, but they organized our own hackathon in Freiburg for the IDR tool. And since that, we've been having regular discussions all the time, chat, GitHub, video calls, wherever. My experience, so I learned from different fields. I got support from all the experts about things that I didn't know before. But I also have to mention that the closed feedback loop was very helpful. So at that time, I was sharing the office with Jean-Karim and E, and that made everything really easy because one can discuss face-to-face, look at the computer together, draw something. This is something that we cannot do anymore, unfortunately. But it was very helpful to have that, and the same for hackathons and any other meetings face-to-face. We also think that this project could have done in collaboration with the IDR and run everything in the EMBL cluster, but we wouldn't have access to the cloud resources if it weren't for EOSC-Life. So that's something that I think it's adding value to this project. And then the other thing I briefly mentioned already, so how we are connecting here Galaxy and the imaging community, and that would work in both directions. On one side, we are bringing Galaxy to the imaging community, that they can combine with other tools, so the imaging people can combine this with other tools for analysis. But at the same time, Galaxy is making these imaging tools accessible to other communities that might need them. Okay, and with this, I would like to thank you all for your attention, but especially Jean-Karim and A for their constant help and support. Jean-Marie and the IDR team for the help from the IDR side, and Bjorn as responsible for the Freiburg Galaxy team and the integration with Galaxy. And with this, I am happy to take questions.